So we did a screen where we generated zebrafish mutants of 10 different autism risk genes, and we analyzed their behavior and using a high-throughput assay, and we looked at their brain structure and their brain activity. And we were interested in identifying potential convergent pathways across these different genes. We found that at a behavioral level, disrupting autism risk genes does lead to alterations in sensory processing and sleep behaviors, but not necessarily in the same direction or to the same extent. So we saw a number of divergent phenotypes at the behavioral level. At a structural level and a brain activity level, we also saw evidence for unique and shared phenotypes across the different mutants. But interestingly, we were able to identify some points of convergence. So for example, we found that the forebrain or telencephalon represents a point of vulnerability to autism risk genes in terms of brain size or brain growth at early developmental stages, whereas we found that the thalamus was one of the most significant contributors to altered baseline brain activity across the different mutants that we studied. So we see very robust phenotypes in our zebrafish mutants of the gene DYRK1A, which has a very conserved effect on brain size all the way down to Drosophila. So it's a Drosophila mini brain gene. And in fact, in zebrafish, we also see a gene dose-dependent effect on brain size, consistent with microcephaly in humans who have DYRK1A mutations. We also see very robust phenotypes associated with a gene called SfN1LAB, which is the zebrafish gene, the equivalent of SfN1A and 2A in mammals.